Welcome back to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID 19. It's Dr. Shikha Agrawal here again, and I'm thrilled you have joined us. In today's episode, we're setting our sights on the virus's most famous feature, the spike protein. Welcome to Target Acquired Unraveling the SARS-CoV-2 spike protein's secret. We are once again joined by Dr. Aiswarya Chaudhuri, expertise in nanomedicine and lipid-based drug delivery systems. Welcome back.
It's great to be back. Thanks for having me again, Dr. Shikha. It's a crucial part of the story.
Indeed, when a new virus like SARS-CoV-2 emerges, it must feel like searching for a needle in a haystack. Where did scientists even begin to look for its vulnerabilities?
Exactly, it's a monumental task. But fortunately, science is often iterative. We have prior knowledge from other coronaviruses, notably the SARS-CoV-1, that is the cause for SARS, and MERS-CoV. Now, when SARS-CoV-2 appeared, its genetic sequence was rapidly determined and shared globally. So this was a game changer. Now, comparisons immediately showed similarities to these earlier coronaviruses, and one protein stood out, that is the spike protein.
Uh, the spike protein, which gives the coronavirus its corona or crown like appearance, right? Why was it such an immediate focus?
Exactly. In other coronaviruses, the spike protein is the key machinery the virus uses to bind to and enter the host cell. Means it's the virus's way of initiating the infection. So the hypothesis was strong from the outset that the SARS-CoV-2 spike protein would play a similar and a critical role. Now, if you block the spike protein, you can potentially block the infection.
That leads really nicely into my next question, Dr. Aishwarya. The spike protein was identified as the main gateway, but it's a complex structure. How did researchers then pinpoint the exact doorknobs or keyholes on this protein that are essential for viral entry?
That's where the multi-pronged scientific investigation unfolded. The first major breakthrough was the identifying the human receptor that the SARS-CoV-2 spike protein uses to enter our cells. Now, drawing from the SARS-CoV-1 playbook, where a protein called ACE2, which the full form is the angiotensin converting enzyme 2, was known to be the receptor. And scientists quickly tested if SARS-CoV-2 also used this ACE2.
Okay. Building on what you just said, did targeting the spike protein prove to be an effective strategy in the development of monoclonal antibodies?
Yes. Now, experiments confirmed that ACE2 was indeed the primary receptor for the SARS-CoV-2. Now, this discovery was pivotal. It meant that the part of the spike protein that directly interacted with the ACE2 was a prime target for neutralization. Now, this specific part is also called the receptor-binding domain, or RBD.
How did they confirm that the RBD was the specific part of the spike making contact with ACE2?
Several sophisticated techniques were employed almost simultaneously. Like structural biology was the absolute key. Using methods like cryo-electron microscopy, X-ray crystallography, the research groups were able to visualize the 3D structure of the spike protein, sometimes even capture in the act of binding to the ACE2 receptor. Now, these atomic level snapshots literally showed the RBD nestled into ACE2, revealing the precise contact points. Now, alongside this, the biochemical and the biophysical assays, such as the surface plasmon resonance and ELISAs, were also used. Now, these lab techniques allowed scientists to produce just the RBD fragment of the spike protein and directly measure its binding affinity to ACE2, confirming its interaction and its strength.
That's fascinating, Dr. Aishwarya. The visual evidence from structural biology combined with direct binding studies painted a clear picture of the RBD ACE2 interaction.
Precisely, Dr. Shikha. Further compelling the evidence came from studying the immune response of people who had recovered from COVID-19. Now, researchers found that many of the powerful neutralizing antibodies these individuals produced were specifically targeting the RBD. This was a strong indicator from the human immune system itself that the RBD is a critical vulnerability.
The RBD sounds like a major bull's eye. Were other areas of the spike protein also identified as important targets?
Yes. While the RBD is a dominant target for many of the most potent neutralizing antibodies, it's not the sole area of interest. Now, another important region within the larger S1 subunit of the spike protein is the N-terminal domain or NTD. Now, antibodies binding to the NTD have also been shown to neutralize the virus. Now, the NTD seems to play roles in early stages of the viral cell interaction, and it has also been a site for frequent mutation in emerging variants.
Well, how were these non-RBD sites like the entity pinpointed as important?
It was often through a process of elimination and broader screening. Now, when isolating antibodies from the recovered patients or developing them in the lab, the scientists would test their ability to neutralize the virus using pseudovirus neutralization assays. Now, in these assays, a harmless virus like VSV or lentivirus is engineered to display the SARS-CoV-2 spike protein on its surface. Now, if an antibody could prevent these pseudovirus from infecting cells, it was considered neutralized. Now, researchers would then perform epitope mapping to determine precisely where on the spike protein these neutralizing antibodies were binding. This meticulous work helps to identify the NTD and other, sometimes more conformation dependent epitopes as relevant targets.
Great explanation, Dr. Aishwarya. Now let's tackle another aspect of this topic. You have mentioned advanced imaging and lab assays. Did computational tools play a part in this rapid identification process?
Absolutely. Computational biology and bioinformatics are crucial accelerators. Now, as soon as the viral genome was available, computational models could predict the 3D structure of the viral proteins like the spike. And these models also help predict which part of the spike were likely exposed to the viral surface and therefore accessible to antibodies, and which region might be involved in interactions like with the human proteins like ACE2. Now, these predictions help to prioritize and guide the wet lab experiments.
It's fascinating how all these different scientific disciplines came together. Now, this detailed understanding of the spike protein's target site, like the RBD, the NTD, how did they directly inform the development of countermeasures like vaccines and monoclonal antibody therapies?
This knowledge was the absolute bedrock. For monoclonal antibody therapies, it allowed the scientists to firstly rapidly isolate and select the most potent neutralizing antibodies from the recovered patients because they knew that these antibodies were likely targeting the RBD on the NTD. And secondly, design and engineer synthetic antibodies are specifically aimed at these vulnerable epitopes. For vaccine development, the vast majority of the COVID-19 vaccines, whether mRNA or viral vector or protein subunit, are designed to elicit an immune response against the spike protein. By presenting the spike protein or the genetic code for it to our immune system, the goal is to generate the neutralizing antibodies that will recognize and bind to these critical sites, particularly the RBD, thereby blocking the viral entry if we are exposed to the actual virus.
It truly highlights how fundamental research paves the way for practical solutions. As always, we want to remind our listeners that this podcast is for informational purposes only and does not constitute medical advice. Any health related questions should be discussed with a qualified healthcare.
That's a very important point to detail it.
The speed at which crucial viral targets were identified and characterized during the pandemic was extraordinary. What do you attribute to that?
It was a combination of factors. Decades of prior research on other coronaviruses and viral entry mechanisms laid the groundwork. Incredible technological advancement in areas like gene sequencing and structural biology allowed for rapid analysis. And critically, an unprecedented level of global scientific collaborations and open data sharing really supercharged the effort.
It's a powerful example of science rising to a global challenge. Thank you once again, Dr. Aishwarya, for these invaluable insights into the detective work behind understanding SARS-CoV-2.
It's been a pleasure to share this, Dr. Shikha.
That wraps up today's conversation. Huge thanks to our listeners for joining this episode of Neutralizing COVID, the monoclonal antibody story. We hope this has given you a deeper appreciation for the intricate science involved in identifying and targeting viral threats. Follow the show for more fascinating insights coming your way. Until next time, take care and keep exploring science.